Today, we are going to look at the digestion of starch by salivary amylase. So, we will first of all start by looking at what starch is. So, when we talk of starch is just a polymer of D-glucose units linked by alpha-1,4 glycosidic bonds, or both alpha-1,4 and alpha-1,6 glycosidic bonds. And starch is made up of two polysaccharides, and these are the amylopeptin and the amylose. So now let me look at the amylose. So amylose are unbranched chain polysaccharides with alpha-1,4 glycosidic bonds. So these are the unbranched chain polysaccharide with the alpha-1,4 glycosidic bonds between the glucose units. So over here we have the alpha-1,4 glycosidic bond. Over here to there's another 1,4 glycosidic bonds formed over here. Amylopeptin. So amylopeptin are branched chain polysaccharides with both alpha-1,4 and alpha-1,6 glycosidic bonds. So as you can see over here, this is the molecular structure for amylopeptins, and these are the bonds. So this is the alpha-1,6 glycosidic bonds over here, and alpha-1,4 glycosidic bond also over here, as shown in the molecular structure. So the next thing to look at is amylase. So first of all, amylase is an enzyme that breaks down or hydrolyzes starch into glucose molecules, which might either be a monosaccharide or maltose, which is a disaccharide. We have three categories of amylases, and these are the alpha amylase, beta amylase, and gamma amylase. And they differ in the way they attack the bonds of the starch molecules. Alpha amylase is widely spread among living organisms, example in the digestive system of human and many other animals. And alpha amylase called the ptyalin is produced by the salivary gland, whereas pancreatic amylase is secreted by the pancreas into the small intestine. And what you have to note is that the optimum pH of alpha amylase is between 6.5 to 6.8. And the salivary amylase is a glucose polymer cleavage enzyme that is produced by the salivary gland. So let us look at some sources of amylase in the human body. So we have the major source and the minor source. So the pancreas and the salivary gland happens to be the major source of amylase in the human body, and the skeletal muscle, the liver, and the fallopian tubes happens to be the minor sources of amylase in the human body. So one key method that we are going to employ in this experiment is the Folin-Wu method. So when we talk of Folin-Wu method, it's just an assay which is used to quantify the amount or the concentration of glucose in a sample. So the Folin-Wu method procedures are: 1, to pipette 1 ml of your sample into the Folin-Wu tube. 2, you add 2 ml of alkaline copper tartrate and place in boiling water about for 8 minutes. 3 is to cool under running water to room temperature. 4 is to add 2 ml of phosphomolebic acid and gently mix. 5 is to make up the 25 ml with distilled water and mix gently. And 6 is to measure and record the absorbance at 580 nanometer. So keynote: for detailed information on the Folin-Wu method, please subscribe to watch my video on Folin-Wu method. I have explained the Folin-Wu method into detail in my video, so please just subscribe to watch. Now, so let us also look at some keynotes in this experiment. So, the test method to determine the digestion of starch by salivary amylase is in two parts. So, the first part happens to be the plotting of the calibration curve using the Folin-Wu method, and the second part happens to be the digestion of starch by salivary amylase and subjecting it to Folin-Wu method. So let's start by looking at the first method, which is the test protocol 1, and which is plotting of calibration curve using the Folin-Wu assay. So the materials or the reagents that we are going to use in this experiment is 1, the glucose standard of concentration 10 milligrams per ml, 0.01 molar phosphate buffer containing 0.5 molar sodium chloride of pH 6.5. We also have our starch of concentration 50 milligrams per ml. We have our trichloroacetic acid, which is 30%. We also have our salivary amylase as our enzyme. We have phospholipid acid. We have alkaline copper tartrate reagent, and we also have our UVV spectrophotometer at wavelength 580 nanometer. So let's start by looking at the methods involved in plotting or preparing a calibration curve. So example, to prepare calibration curve from 0.0 to 2.0 milligrams per ml in 6 Folin-Wu tube from our standard, which is 10 milligrams per ml glucose standard over here. Now, so the first thing to do is to set up a wall label 6 Folin-Wu tube. And the next thing to do is to prepare a series of dilution of concentration ranging from 0.0 to 2.0 milligrams per ml from our glucose standard. So as you can see over here, the various concentrations that we are going to prepare to plot the calibration curve are 0.0 milligrams per ml, 0.4 milligrams per ml, 0.8 milligram per ml, 1.2 milligrams per ml, 1.6 and 2.0 milligrams per ml from the 10 milligrams per ml glucose standard. So let's just see an example of how to prepare these various concentrations from our 10 milligrams per ml glucose standard. So example, so using direct dilution to prepare a final volume of 5 ml for the 0.0 milligrams per ml, 0.4 milligrams per ml, and 0.8 milligrams per ml from the standard glucose of concentration 10 milligrams per ml. Yeah, so in this video, I just did the calculation for these three various concentrations, so 0.0, 0.4, and 0.8 milligrams per ml. So I will also advise that you do for the 1.2 milligrams per ml, 1.6 milligrams per ml, and 2.0 milligrams per ml using the same procedure that I will use over here. So let's start from the 0.0 milligrams per ml. So we are going to use the equation C1V1 equals C2V2. So where our C1 is the standard concentration, which is the 10 milligrams per ml. Our C2 is the final concentration here, which is the 0.0 milligrams per ml. V2 is the final volume, and it's going to run through from the example given. And V1 is the volume of standard needed. So making V1 the subject, we have V1 equals C2V2 all divided by C1. So if you are to substitute these values into this equation, you get V1 to be equal to 0 mils. So let's look at the 0.4 milligrams per ml. So how to prepare this concentration from our stock solution using the direct dilution. So also using the equation C1, V1 equals C2V2, as explained earlier, C1 is a standard concentration, C2 final concentration, and the final concentration over here is a 0.4 milligrams per mole because this is the concentration that we are going to prepare over here. We have V2 to be our final volume, which is 5 mils, and V1 is the volume of the standard glucose needed to prepare this concentration in a final volume of 5 mils. So we make V1 the subject. So making V1 the subject, we have V1 equals C2V2 all divided by C1. And if we are to substitute our values over here into the equation, we will get our V1 to be 0.2 mils. So the next concentration that we are going to prepare is 0.8 milligrams per mole. So also we will use the equation C1, V1 equals C2, V2, where C1 is a standard concentration of the glucose, C2 our final concentration, which is a 0.8 milligrams per ml over here. V2 is our final volume, and V1 happens to be the volume of our standard needed to prepare the final concentration 0.8 milligrams per mils in 5 mils. So making V1 the subject, we get V1 equals C2V2 divided by C1. So substituting these values to the equation, we'll get V1 to be 0.4 mils. So I will encourage you to do for the 1.2 milligrams per mil, 1.6 and 2.0 milligrams per mil. Yeah, so this is just a table that I have done here to explain the calculations that I have done for you. Now, so we can see that from the table, we have the tube numbers labeled as 1 to 6 with the concentrations in milligrams per mil in each of the test tubes. So we have 0.0 to 2.0 milligrams per ml over here. So from the calculation, these are the volume of glucose or the standard glucose needed and the volume of distilled water needed in mils to prepare these concentrations over here. So from the calculation, we saw that the V1 for the 0.0 milligrams per ml is 0.0. So to prepare this concentration, we just pick 0.0 mils and add it to 5 mils of distilled water. So to prepare the 0.4 milligrams per ml, so we'll pick 0.2 mils of our standard and add it to 4.8 mils of distilled water. So to prepare a concentration of 0.8, we pick 0.4 mils of the glucose standard and add 4.6 mils of distilled water to it to prepare this concentration. So to prepare this concentration, we pick 0.6 mils of our glucose standard and add 4.4 mils of distilled water to it. To prepare this concentration, we pick 0.8 mils of our glucose standard and add 4.2 mils of distilled water to it. And finally, to prepare this concentration, we pick 1 mil of our glucose standard and add 4 mils of distilled water to it. So after preparing our glucose standard, we now pipette 1 ml of each sample. So that's a prepared glucose standard into its respective tube. And after that, we add 2 ml of alkaline copper tartrate and place in a boiling water bath for 8 minutes and cool under running water to room temperature. And what you also have to note is that the alkaline copper tartrate makes available the cupric ions and make the medium alkaline, and the temperature aids to facilitate the reaction, which is the reduction of the cupric ions to cuprous oxide. So the next is to add 5 ml of phosphomolebic acid and gently mix. And what we also have to note is that the phosphomolebic acid reacts with the cuprous oxide to form molybdenum blue, so which is a blue solution. The next step is to make up to the 25 ml with distilled water and mix gently. And the next step is to measure and record the absorbance at 580 nanometer. So what I can say is that from step 3 to step 7 happens to be the Folin-Wu method. So this is the assumed table of result. It is an assumed table of result. Yeah, so from the table we can see we have the number of tubes over here, the glucose concentration, and this is the volume of glucose needed and the volume of distilled water needed to prepare the various concentration over here. And this is the measured this absorbance value at 580 for each of the concentrations. So for this concentration we have this. For this concentration we have this. For this concentration we also have this value. For this concentration we have this value. For this concentration we have this value. And for this concentration we also have this value as seen over here. So the next thing to do is to plot the calibration curve by plotting the absorbance against the glucose concentration. So the absorbance is on the y-axis and the glucose concentration is on the x-axis. So this is the nature of graph that we are going to get as our calibration curve. And this is our line of equation giving us y equals 1.0331 s, where y is the absorbance, s is the slope, where y is the absorbance, 1.0331 is the slope, and s is the glucose concentration. And this is the r squared giving us 0.9941 over here. So this is the calibration curve. And one keynote is that for detailed information on calibration curve, please subscribe to watch my video on calibration curve. So the next thing to do will be the digestion of starch by salivary amylase and subjecting it to the Folin-Wu method. So this is the second protocol or the second part of the experiment. Now, so in this part, the first thing to do is to prepare a substrate buffer. Okay, so the first step says preparation of substrate buffer by adding 5 ml of our starch solution to 5 ml of our 0.01 molar phosphate buffer containing 0.5 molar sodium chloride at pH 6.5. And what we also have to note is that the 0.5 molar sodium chloride in the buffer is acting as an activator, or is going to act as an activator for our enzyme that will be introduced into the substrate buffer. So the next is to equilibrate the substrate buffer and the enzyme at 37 degrees Celsius in a water bath for 5 minutes. So as you can see, this is our water bath set to 37 degrees Celsius, and this is our enzyme in this test tube, and this is also our substrate buffer of final volume 10 ml in the water bath undergoing equilibration at 37 degrees Celsius for 5 minutes. So the next step is to perform an enzyme substrate reaction by adding 1 ml of the equilibrated enzyme to our substrate buffer in the water bath at 37 degrees Celsius. So what you also have to note is that we are supposed to leave the enzyme substrate reaction to stand in the water bath at 37 degrees Celsius. So the fourth step is to stop the enzyme activity with 0.1 ml 30% TCA at different times intervals. So these are the times intervals: 0 minute, 2 minutes, 5 minutes, 10, 30, and 60 minutes. And over here too, there are some keynotes that we have to know. We are supposed to leave the enzyme substrate reaction to stand in the water bath during these time periods over here. and also all the 6 test tubes over here contains 0.1 ml of 30% TCA. So what it means is that before you pick 1 ml of your substrate enzyme reaction into your test tube, you must first put 0.1 ml of 30% TCA into each of the test tube before you add your enzyme substrate reaction to it to stop the reaction. So as you can see over here, this is our substrate enzyme reaction placed in a water bath at 37 degrees Celsius. And these are the various test tubes that we have over here. So at 0 minutes, we pick 1 ml of our substrate enzyme reaction and add it to a test tube containing 0.1 ml 30% at 0 minutes. So at 2 minutes, we pick 1 ml of the enzyme substrate reaction and add it to a test tube containing 0.1 ml of 30% TCA. At 5 minutes, we do the same. At 10 minutes, we do the same. At 30 minutes, we do the same. And at 60 minutes, we do the same as seen over here. So after stopping the reaction, we now subject the stopped reaction to Folin-Wu method or Folin-Wu assay. And we do this by pipetting 1 ml of the stopped reaction, so which is now our sample, into a Folin-Wu tube. The next is that we add 2 ml of alkaline copper tartrate and place in boiling water bath for 8 minutes. We then cool under running water to room temperature and add 2 ml of phospholipid acid and gently mix. We then make up to the 25 ml mark with distilled water and mix gently. And finally measure and record the absorbance at 580 nanometer. So these are our assumed absorbance values at 580 nanometer after we subject the stopped reaction to the Folin-Wu method over here. So as we can see, at 0 minutes, we had an absorbance of 0.00. At 2 minutes, we had 0.016. At 5 minutes, 0.710. At 10 minutes, 0.880. At 30 minutes, 0.970. And at 60 minutes, 1.880 as the respective absorbance values for each of the time. So the next thing to do is to interpolate or extrapolate the glucose concentration from the calibration curve. So what we first have to do is to use a calibration curve given over here to extrapolate or determine the concentration of the glucose hydrolyzed from the starch by the enzyme amylase. So what you can see over here is that we have our calibration curve with the equation of line giving us y equals 1.0331s. So where y happens to be the absorbance value obtained and s happens to be the concentration of the glucose, and the 1.0331 happens to be the slope or the gradient of the graph. Yeah, so to extrapolate the glucose concentration from the calibration curve, we will have to use the equation of the line given as y equals 1.0321s and make s the subject. So making s the subject, from this equation we have s equals y divided by the slope, which is 1.0331 over here. So what we are going to do is that we are going to use the absorbance values from the table to calculate for the concentration. So we know that at y equals 0, that is from the absorbance table, our S will be 0.00, because if we are to substitute um y as 0 into the equation, we will get s to be 0, 0. So at time 2 minutes, we had an absorbance value of 0.016 from the table. So if you are to substitute this value into the equation, s equals y divided by 1.0331, we will get an s value to be 0.015. So at 5 minutes, we had an absorbance value of 0.710. So if you have to substitute this into the equation, we will get an S value to be 0.687. At 10 minutes, we had an absorbance value of 0.880. And if you have to substitute this into the equation, we will get an S value of 0.852. At 30 minutes, we had an absorbance value of 0.970. And if you are to substitute this into the equation, we will get an S value to be 0.939. And at 60 minutes, we had an absorbance value of 1.880. And if you have to substitute this into the equation, we will get the S value to be 1.820. So the S values obtained here happens to be the extrapolated glucose concentration from the calibration curve. So the next is to calculate the percentage hydrolysis, and we do this by assuming that 60 minutes gives a complete hydrolysis, which is equal to 100%. So from the table, so we can say that at 60 minutes, the extrapolated glucose concentration is given as 1.820, and this is equal to 100. So this concentration is equal to 100%. So what happens is that to calculate the percentage hydrolysis at 0 minutes, we will have the glucose concentration at this minute divided by the glucose concentration at 60 minutes times 100. So as you can see over here, we have 0.00 divided by the 1.820 times 100 to give us 0.00. So the percentage hydrolysis at 2 minutes will be this value, 0.015 divided by the 1.820 times 100, and to give us 0.82. The percentage hydrolysis at 5 minutes will also be the concentration 0.687 divided by this times 100, and we do that, we'll get 37.75. And the percentage hydrolysis at 10 minutes will be the concentration over here divided by this times 100, and we'll get 46.81. And the percentage hydrolysis at 60 minutes will be this concentration divided by this times 100 to give us 51.59. And the percentage hydrolysis at 60 minutes will obviously be 100, because we are assuming that at 60 minutes there is a complete hydrolysis, so which is equal to 100%. So this is a table of results showing um, the various times with their respective glucose concentration from the calibration curve and the calculated percentage hydrolysis right here. So the next thing to do is to plot a graph of the percentage hydrolysis against time. So we do this by plotting the percentage hydrolysis on the y-axis and the time in minutes on the x-axis. So this is our time in minutes, and this is our calculated percentage hydrolysis. So if you are to plot these values against this will be the nature of the graph that we will get over here. And the graph happens to be a graph of percentage hydrolysis against time, as seen over here. Nice.